We're going to switch gears now and start talking about the host cells that viruses infect. It turns out that all life on this planet, all types of cells and multicellular organisms can be infected by viruses. It doesn't matter if it's a bacterial organism or an archaeal organism or a eukaryotic organism. There are viruses that are capable of infecting it. Viruses, it turns out, are host specific. In other words, each type of virus has a particular host cell or host organism that it infects. But, and this is important, host specificity can change over time, and it changes through mutation. Remember, viruses are obligate intracellular parasites. They cannot exist without a host cell to replicate them. If a viral particle cannot find a host cell to get into, that viral particle will eventually degrade and disappear. It needs the host cell to make copies of it. It's the host cell that does the work of reproducing the virus. This requires the viral particle then to get in to the host cell that is going to replicate it. Host cells, therefore, have to have some entryway for the viral particle to pass through. And that entryway is some kind of a receptor on the surface of the host cell membrane. Now, it's interesting to note that some viruses can only bind to receptors on one type of cell inside the host. So obviously I'm talking about multicellular organisms here. Influenza is only capable of binding to a particular receptor on one type of cell in our respiratory tract. But there are other viruses that bind to receptors that are found on multiple types of cells. Epstein-Barr is like that. So some viruses can only get into one kind of cell inside their host, and other viruses can get into multiple types of cells in their host. Think about the SARS-CoV-2 virus for a minute. I told you earlier that the SARS-CoV-2 virus binds to a receptor called ACE2. ACE2 stands for angiotensin converting enzyme 2. Now, this is an enzyme that is involved in changing how narrow or how wide our blood vessels are. So it comes into play with your blood pressure. The ACE2 receptor is widely distributed throughout the human body. It's found on the endothelial cells. Again, those are the specialized epithelial cells that line the blood vessels. It's also found on the epithelial cells that line the alveoli inside your lungs. It's found on the smooth muscle cells that are part of your arteries. It's found on the cells that line your small intestine. It is widely distributed, and that's why you can see a range of signs and symptoms from people who get infected with SARS-CoV-2 and develop COVID-19. Yes, we first contract this virus through the respiratory tract, and it first gains entry through those alveolar cells. But it then gets carried throughout other parts of the body, and it can bind to any cell that has that ACE2 receptor on it. So SARS-CoV-2 is similar to the Epstein-Barr virus in that it can bind to multiple cell types, because in the human body, multiple types of cells express the ACE2 receptor on their surface. This slide shows us a colorized EM image of SARS-CoV-2 over on the left-hand side. There have been lots and lots of EM images of this virus available to us online since the start of the pandemic. You can really see the spike proteins around the edge of this viral particle. I like this particular image because it helps us see why somebody called this a coronavirus. A scientist looking at this virus under the electron microscope saw these spike proteins and thought, you know what, this looks like a crown. It looks like this cell is wearing a crown, and hence the corona name. If you look at the image on the right-hand side, you can see a depiction of how this virus gets into our cells. Here's the drawing of the SARS-CoV-2 virus, again with its genetic material and its capsid, the envelope, and then these pink spike proteins. The spike protein is able to bind to the ACE2 receptor. And in this drawing, the ACE2 receptor is sitting in the membrane of the cell. So the human cell here is blue on the inside, and the environment is out here where you see this beige color. The virus comes in, it binds to the ACE2 receptor, and a change occurs. That change is that the membrane is now going to start to invaginate. It's going to bend inwards, allowing this viral particle to gain entry into the host cell. We said that viruses are host specific. They infect one kind of host, either a host cell or a host organism. But we also said that that specificity can change over time when the genetic material mutates. It's important to know that this kind of a mutation is relatively uncommon. Viral genetic material itself is going to mutate fairly frequently. Remember the example of influenza. But most of those mutations are not going to cause a change in host specificity. The kind of mutation that would allow a virus to change hosts, in other words, to go from one type of host to another type of host, that kind of mutation is actually uncommon. It's also important to know that mutations are random. So it's not that the virus can choose to mutate and change hosts. It has to just exist. And every once in a while, a mutation will happen that allows it to change hosts. Now, mutations are fairly common, but they are more common in some viruses than others. The influenza virus, as we said, mutates all the time, sometimes several times a year. SARS-CoV-2 seems to mutate a little more slowly, although as long as it is a pandemic virus, in other words, as long as it is circulating around the globe, it means that millions and millions of people are infected, and millions and billions of copies of the virus are being made every day. And the enzyme that is responsible for copying the genetic material is making mistakes and creating mutations. Remember, mutations are a natural thing. You don't have to get exposed to some strange chemical or radiation to have your genetic material undergo a mutation. The enzyme that copies genetic material, the enzymes that copy genetic material make mistakes. They are biological molecules. They are not perfect. Mutations occur in nature all the time. It's just that sometimes mutations give viruses new traits that are dangerous to us. Sometimes when a virus mutates, it has the ability to infect a new kind of cell. Sometimes it can infect a brand new host. These types of big events, big mutation events, are not very common, but they do occur. We know, for example, that the SARS-CoV-2 virus most likely was the result of a mutation in a bat coronavirus. And I have the name of that bat coronavirus right here. It's quite a long name. It's SARS-R, RA, bat-CoV, RATG13. This is the coronavirus that has genetic material, RNA, that is most closely aligned with SARS-CoV-2. In fact, when you compare the RNA inside this bat virus with the RNA inside SARS-CoV-2, there's a 96% overlap. In other words, 96% of the bases are exactly the same in these two viruses. There's only a 4% sequence difference. And that's why most scientists believe that there was a mutation event that allowed this bat virus to now infect human beings and specifically bind to the ACE2 receptor. Sometimes mutations create these big jumps, like infecting new cells or infecting new hosts. But sometimes mutations make smaller changes. For example, sometimes the host will be exactly the same, the cell will be exactly the same, but the mutation will allow the virus to bind to a new receptor. Or maybe bind to the same receptor type, but more effectively, more tightly. A good example of this is in avian influenza, or what we sometimes refer to as bird flu. Influenza viruses mutate a lot. And this type of influenza has birds as its natural host, but it's been mutating. And recent mutations have allowed this type of influenza to switch from a bird host to a human host. Now, here's the good news for us, because avian influenza is a particularly bad pathogen. While it has now developed the ability to sometimes pass from birds to humans, it doesn't yet have the ability to be transmitted from one human to another human. That's good news for you and I. There may come a day, though, that the avian influenza virus gains that trait and is able not only to pass into humans, but to pass from human to human. That is, at least potentially, going to produce another pandemic. Now, when is that likely to happen? The answer is no one knows. And that's because mutations are random. That kind of a mutation could happen tomorrow. It could happen 1,000 years from now. And it could never happen. We simply don't know. So once a virus gets into the host that it is able to infect, it has to go through a series of steps in order to use that host cell to make more of itself. The very first thing that has to happen is the viral particle must bind to a specific kind of receptor on the surface of the host cell. This is the single most important step. The viral particle must be able to bind to that receptor, that protein that's on the surface of the host cell, or else it cannot get into the host cell. When the viral particle binds to the receptor, what's going to happen is the receptor, remember the host receptor, is going to change shape. Essentially what's happening is the viral protein, often that spike protein, and the host receptor protein are attaching to each other and creating a brand new protein. And of course, anytime you change a protein, you're going to change the way it folds or the shape that it takes. So the binding of the viral particle to the host receptor creates a shape change, and that's going to allow either the entire viral particle or at least the genetic material to come into the host cell. The virus is essentially going to disassemble once it's inside the host, and host enzymes are going to go to work doing what they do inside the host cell. They're going to replicate the viral genetic material, and they're going to transcribe and translate viral proteins. Remember earlier when we were talking about viral shape, and specifically complex shape, like bacteriophages, we said that sometimes host cells use their own chaperone proteins to help build the newly made viral particles. But other times, viral proteins can just self-assemble because they're getting into such a simple shape, something like a polyhedron or a helix. Once the new viral particles are assembled, now it's time for them to exit the host cell, and there they will go on to infect other cells. Retroviruses are one of the most difficult viruses to deal with in medicine. Retroviruses, by definition, are RNA viruses that are capable of inserting a DNA copy of their genetic material into the host cell's DNA. And the most famous example of this is the human immunodeficiency virus, or HIV. This is the virus that uses this enzyme called reverse transcriptase. There are other viruses too that have now been discovered that have this enzyme. Remember the central dogma. Remember that normally in a living cell, DNA is used to make RNA through transcription. But the viruses that have reverse transcriptase can do the exact opposite. The reverse transcriptase enzyme can take an RNA molecule and make DNA from it. So what the human immunodeficiency virus does when it gets into the human host cell is it uses reverse transcriptase to take its RNA genome and turn it into a DNA molecule. Then that DNA molecule is inserted into the host cell's DNA, and there it will sit. Now, that host cell, which happens to be an immune cell called a T cell, that host cell is going to go on and live its life as though nothing has happened. It's going to divide at a certain time interval, making new T cells, and every time it divides, it's going to make a copy of its own DNA. Because it's copying its own DNA, it's also copying that viral DNA that has been tucked inside of it. The other thing that's happening is the T cell is using its DNA to make protein, just like any cell does. But because the viral DNA is tucked in there, it's also making viral proteins. Reverse transcriptase is a viral enzyme. It gets packed into the virion, pre-made, pre-assembled, pre-built, so that as soon as the viral particle gets in to the host cell, it can start doing this reverse transcription. We now know that the human immunodeficiency virus can insert its DNA into a host cell and sit there for as long as 10 years before it finally comes back out, gets packaged up into new viral particles, and exits the cell. This is a terrible, terrible trait of this virus, because people who are infected with HIV can go for 10 years and not know it, go without any signs of AIDS. And of course, during that time, they can pass the virus to susceptible partners.